Hello, students. Today we have to discuss the complement system, its activation, and its role. So, the first thing is that what is complement system? The complement system is a system of proteins, about 30 proteins, which are taking part in the immune system or immunity, or you can say it is a component of the innate immunity. These 30 proteins, they are named as C1, C2, C3, C4, C5, C6, C7, C8, C9. So these proteins are basically present in our blood, in the serum, in inactive form. But when the threat will come, when the pathogen will come, when the bacteria or viruses will come, then it will become active. So, and then it will help to defend our body and will help to mark the target and will help the immune system to identify the target and to eliminate the target. So here you can see this. There are three different pathways for activation of complement system. That is classical pathway. So this classical pathway and alternative pathway and lectin pathway. So there are three pathways for the complement activation. Classical pathway or antibody-mediated pathway and lectin pathway and alternative pathway. So in the most important one is the antibody-mediated pathway or classical pathway in which this pathogen will enter, and when it will enter, there will be some antibodies already there against this pathogen. And these antibodies will bind, will recognize and will bind with the, will recognize and will bind with the pathogen. And then in the FC part, there will be some changes. And the FC parts, when there will be some changes in the FC part, then it will C1, Q2, R2, they will come and bind here. And then this C1 R and C1S, they will bind with C1Q, and this complex will bind with the FC part of the antibody, which is in the bound form with the bacteria or virus. And then this complex will act as an enzyme, and then it will result in the cleavage of C2 and C4. And when this cleavage of C2 and C4 will take place, so when this C2 cleavage will take place, then it will make C2A and C2B. C2A and C2B formation will take place. So, and the C4 cleavage will take place and it will make C4A and C4B. So these two components, that is C2A and C4B, C2A and C4B, they will come together and they will make a new complex, new enzyme, new protein, active form of the protein. And this C2A, C4B will come and then it will act as a C3 convertase. So it will act on the C3 protein and then it will cleave it in two parts, that is C3A and C3B. So then this C3A and C3B, C3A will act as a anaphylatoxin, and this C3B will come and it will combine with this C2A, C4B, and will make this C4B and 2A and C3B, and it will make another enzyme that is C5 convertase. So this C5 convertase will act on C5, and then it will make this C5A and C5B. C5A will help in the inflammatory response, and this C5B, then this C5B will combine with this C3, C6, C7, C8, C9, and then it will make this membrane activation complex, MAC. And this MAC, this membrane activation complex or MAC, it will help to generate pores in the target cells and will result in its lysis. So the first pathway was, as we discussed, classical pathway that the pathogen will enter, and then this antibodies against that pathogen, which are already there, they will combine, and there will be some change on the FC part of the antibodies, which will combine with this C1Q and C1R and C1S, and then they will act as enzyme and they will act on the C2 and C4, and they will cleave them and will make it C2A and C2B and C4A and C4B. So this C4B and C2A, it will combine and they will make a new enzyme, that is C3 convertase, and C3 convertase will act on the C3 and will make C3A and C3B, and this C3B will combine with C4B and 2 and C3B. They will combine and they will make a new enzyme, that is C5 convertase, which will act on C5 and will make its two pieces, C5A and C5B, and this C5B will come and it will combine with this C6, C7, C8, C9, and will make membrane activation complex, a tubular structure, which will create pores in the target cell. And of course, the target cell can be the virus or the bacteria, and then it will result in the death of that pathogen. Another pathway is that this the pathogen will lectin pathway. In the lectin pathway, this pathogen will be recognized, and this will have this lectin membrane associated lectins, and they will activate this MASP1 and MASP2, and then it will act as this to act on C2A and C2B, and then when the cleavage of C2 and C4 will take place, then it will result in the formation of C3, C2A and C2B and C4A and C4B. And these, just like in the classical pathway, it will make this C4B, C2A, and this C4B, C2A, they will act as C3 convertase and will act on the C3 and will make C3A and C3B, and this C3B will combine with the C4B and C2A, and it will make C3 convertase, C5 convertase, sorry, C5 convertase. And this C5 convertase will act on the C5 and will make this C5A and C5B, and C5B will combine with these C6, C7, C8, C9, and it will make this membrane activation complex. And this membrane activation complex will attack on the target cell and will result in the lysis. So the second path is the lectin-mediated pathway. The other one is this alternative pathway, which can be spontaneous, and in it, there's C3b and capital B, small b, they will combine. It will make the C3 convertase and C3 convertase. Then it will combine, it will make C3a and C3b, and C3b will combine with capital B, small b, and C3b, and will make C5 convertase. So this C5 convertase, then it will act on a C5 and will make C3, this C5A and C5B, and this C5B will come, and then it will bind with C6, C7, C8, C9, and then it will make this. So it will make this. So it will make membrane activation complex, and then it will result in the lysis of the target cell. So, so here you can see that these three different ways will help in the activation of complement system, and then it will help in the elimination, targeting and elimination, and opsonization and neutralization, whatever the case may be. So it also help in the cleaning of the body from these immune complexes. So anyway, so this is all about the activation of the complement system. So see you then in the next lecture.